This method can help answer key questions in the neurodegenerative disease field, such as what genetic factors and pathways regulate the toxicity of disease-associated proteins. The main advantage of this technique is that the highly efficient process of yeast mating is used to screen yeast models against a large collection of library genes.
Though this method can provide insight into the toxicity of neurodegenerative disease proteins, it can also be applied to the study of other cellular processes in yeast, such as tolerance of other stress conditions.
Begin this procedure by aliquoting 5 microliters per well of plasmid DNA from an arrayed plasmid library to round-bottom 96 well plates. Keep the plates at 4 degrees Celsius. Next, inoculate 150 milliliters of yeast peptone dextrose or YPD medium in a 500 milliliter flask with a colony of the haploid yeast strain W303 alpha. Incubate the culture at 30 degrees Celsius with shaking at 200 RPM overnight. On the following morning, after measuring the OD600 of the overnight culture, dilute the culture to an OD600 of 0.1 in 2 liters of YPD. Incubate at 30 degrees Celsius with shaking at 200 RPM for about 5 hours, until the culture reaches an OD600 of 0.4 to 0.6. To harvest the yeast culture, fill 8 sterile 250 milliliter centrifuge bottles and centrifuge at 3000 times G for 10 minutes at room temperature. Pour off the supernatant without disrupting the pellet. Wash the yeast as described in the text protocol and consolidate the cells in